Thank you, Jill, for the introduction, and good morning good afternoon, actually, to everyone. I'm really happy to walk you through to one of the most important stories in the modern medicine, which is the evolution of cancer care. And I would like to start with a story about two women. So let's imagine we are in the 1999, and Maria, 48 years old, she was diagnosed with an aggressive breast cancer, and her oncologist prescribed her like standard chemotherapy. No molecular testing was performed, and the HER2 status was not checked. So the trastuzumab, the drug that could cure her, existed, but she never received it. Now, let's move forward in 2024, and Elena, 45 years old, she was diagnosed with the same type of cancer as Maria, so aggressive HER2 breast cancer, but her tumor was profiled at the diagnosis, and the HER2 amplification was confirmed. So she received the correct therapy that was made of trastuzumab plus pertuzumab and chemotherapy. And the treatment response was monitored through the use of circulating tumor DNA. And Elena was able to reach a complete pathological response. So we are talking about the same disease, the same drug available. The only difference was a $200 biomarker test. So this story illustrates us as why precision oncology matters and why we need to keep pushing the boundaries of cancer diagnostic, which also brings us to the core of the problem. So why 90% of oncology drug clinical trial fails? So if we look back of 10 years, we can see that basically only 3.4% of the overall drug were approved in FDA. And for each drug that fails, the cost for the company was between 1 to 2 billion between R&D cost and development cost. And the reason for the failure of this drug approval in 50% of the case is due to lack of efficacy. But is really the drug that is lack efficacy, or we simply tested in the wrong patient? And so basically, this is the most important concept of precision oncology. So let's take this example. So we are talking about lung cancer, and we know that EGFR inhibitor can basically reach a response rate to therapy of 70% only in patients that present an EGFR mutation. But this mutation is actually present in only 50% of the population. So if we select the population at early of the trials, we can reach this 70% response. The trial succeed and the FDA approve the drug. But if we do not select the population, what happens is that the 15% of patients that present that mutation and that benefit from that drug will be diluted in the overall cohort, which means that the overall response rate to therapy drop off to 10%. The drug seems to be ineffective and the trial fails. So the take-home message in this case is that the drug actually worked, but we just tested in the wrong patients. And so from here, the central dogma of precision oncology, which is the right drug in the right patients at the right time. So the era of precision oncology started with the very first milestone only in 1998, where trastuzumab, the drug we were talking about, Maria and Elena, was approved. This drug was effective only in patients that present this HER2 amplification, and for the first time the FDA required a diagnostic test before prescribing a cancer drug. Just moving forward 3 years later, we came out with the imatinib that turned the leukemia from a death sentence to a manageable chronic disease with a 98% response rate by targeting the BCR-ABL fusion proteins. And then another great milestone in precision oncology area was the 2017 with the release of pembrolizumab. This was the first FDA-approved tissue-agnostic therapy, which means that the tumor was profiled for mutational burden, and if the patient has a high mutational burden, it has the 40% probability to respond to this type of therapy. This was revolutionary because basically for the first time, we were targeting cancer by its molecular profiling and not by the organ it was growing in. And then we have 2025, in which there was the first AI-based prognostic tool for prostate cancer treatment personalization, showing the convergence between biomarker and artificial intelligence. However, we don't need to forget that all the new biomarker discovery and targeted therapy that are described in here can be possible and are supported only by technology advancement. So also in technology development, we had important milestones that helped the development of biomarker and precision oncology journey. For instance, in the early of the 80s, there is the invention of a PCR, and at the beginning of 90s, we have the commercialization of the first automated sequencer, and the omic term was finally introduced. 2003 is another pillar of the technology development, because the Human Genome Project was completed. So the whole human genome was sequenced and mapped. And in 2010, we started with the concept of integrative multiomic and single cell sequencing. And finally, in the last 10 years, from 2016, we have the boost in machine learning and artificial intelligence field. And the numbers are also clear, because thanks to precision oncology, now we have more than 70 FDA-approved targeted therapy. And trials that are guided by biomarker selection require 5 to 10 times less patients compared to normal trial. And up to date, 90% of phase 3 clinical trial use biomarker for patient stratification. So this brings us to the new era. So of biomarker, we've established the problem and the solution in principle, and now we will dive into the technologies that make this possible, starting with the biomarker classification. So biomarkers have different purpose depending on where the patient is within his disease journey. So we can have risk biomarkers that basically assess the probability that the cancer will develop. Then we have diagnostic biomarkers that confirm the presence of the disease. Prognostic biomarker instead predict how the disease will behave regardless of the treatment. And one of the best examples are Oncotype DX, which is a 21 gene expression test that helped more than 70% of breast cancer patients avoiding unnecessary chemotherapy. And then we come to the cruel jewels of the precision oncology, which is the predictive biomarkers. Basically, they tell you which drug will work, and this is the case of HER2 that predicts the trastuzumab response, or EGFR that predicts the tyrosine kinase inhibitor response. Finally, we have pharmacodynamic biomarker that confirms that your drug is actually hitting its target, and MRD, so minimal residual disease, or monitoring biomarkers that track whether cancer is truly gone after the treatment. So the molecular toolkit for cancer biomarkers spans in the 5 major classes. So we have protein, metabolites, DNA with its mutation, combinable alteration, and epigenetics, so methylation pattern, and also the analysis of cell-free DNA fragments. And then finally, we have the RNA with the transcriptome profiling, gene fusion, and expression signature. But we've evolved from a single protein to a single gene to a panel of hundreds of genes to multi-omic integration between DNA, RNA, and proteomic. And right now also powered by the use of novel artificial intelligence tools. And the data also show the transformation in trial design. So if we look at this chart, we can see that basically in 25 years, we passed from the only 15% of the trial that were using biomarker-driven patient selection to the 2025, in which we can see that more than 73% of the clinical trial are based on biomarker-driven patient selection. So in 25 years, we had a 5x growth with more than 50,000 cumulative biomarkers that guided this trial. And also the inflection points are clear. So we passed from the single gene area that gave wave to the NGS panel and basket trials in 2012. And the current wave of multi-omics and AI that is accelerating the adoption is even farther. But all of these rely on one thing, so getting tumor molecular information. And that brings us to a critical question. So which type of tissue do we use for getting this critical information? So solid or liquid biopsies? In my opinion, tissue and liquid biopsies are complementary approach, not replacement. So the tissue is still gold standard for histopathological confirmation and can give us also information regarding genomic profile of the tumor, while of course the histopathological confirmation is not possible with liquid biopsies. But however, liquid biopsies wins on invasiveness because it's just a blood draw versus a surgery. It wins on tumor coverage because it is capturing the overall biomarkers from tumor sites versus just one piece of the biopsies. It wins also on repeatability because theoretically it can be repeated weekly or monthly, while the surgery is limited for the acquisition of tissues. And also the turnaround time is usually lower in liquid biopsies compared to tissue biopsies that span between 7 to 14 days according to the type of test that we are trying to perform. So if it is based on a PCR approach or on an NGS approach, while for the tissue biopsy, the turnaround time is averagely around 1 month. But the tissue biopsy still wins on sensitivity, especially for low shedding tumors. However, as reported by different open database for approved diagnostic tests, we can see that nowadays, tumor genomic profiling is equally distributed between tissue and liquid biopsies. And among the liquid biopsies, what is mainly analyzed are the cell-free DNA that represent the 60% of the total approved test, followed by this multi-omic approach, while the RNA represents just the 8% of the overall. And if we look at the application of this cell-free DNA, we can see that there are several clinical applications across the cancer care, and for sure the first one is as a companion diagnostic, and we can mention, for instance, the COBAS EGFR mutation that was the first FDA approved liquid biopsies companion diagnostic in 2016. Another important application are actually monitoring biomarker and minimal residual disease, such as that helps to find a microscopic residual disease. And we can mention a few tests such as the one from Signatera that was approved just a couple of months ago and has application for breast cancer, colorectal cancer, and ovarian cancer with a sensitivity of 94% and specificity of 99%. And Guardant 360 Liquid with a specificity of 99% that can be applied to all type of solid tumor. So for monitoring biomarkers, cell-free DNA are quite used, especially because since they are detected in liquid biopsies, it is easier to repeat it during treatment of the patients. And finally, we have also some application as early detection. And we can mention the Gallery test powered by Grail, that is a multi-cancer screening via methylation pattern, in which they can screen more than 50 types of different cancers with a sensitivity of 67% and a specificity of 99.5%. But DNA doesn't tell us the whole story. We also have RNA that is used in clinical setting as biomarkers. So if we have to mention, we have the Colosense, which was the first FDA-approved RNA-based biomarker. And this was approved for the diagnosis of colorectal cancer. While a different approach is a liquid hallmark that basically put together 80 signatures from cell-free DNA and 10 signatures from cell-free RNA. The combo of cell-free DNA and cell-free RNA brings to almost 40% increased detection compared to the cfDNA alone. And this is an NGS-based diagnostic test approach. So the comparison between cfRNA and cfDNA is basically clear. I think that cfRNA can give us a better insight into the gene expression due to their diversity and dynamic natures, and so they have a huge potential as biomarker and also as a less confounding effect with cell death signature compared to the cfDNA. But we don't, when we talk about RNA profiling, we don't need to think only to mRNA signature or expression, because the central dogma of biology tells us that all phenotypic information must pass through the translation. So translation is actually the key regulatory node in gene expression and governs the protein production in response to different physiological conditions. But so when we talk about translation, we are talking to different kind of RNA molecules that are involved. So apart from the mRNA that represent basically the textbook that must be read by the ribosome, that is actually the core machinery of the whole process. And then we have the tRNAs that are the adapter molecules that basically read the codon triplet from the mRNA and it translated into an amino acid code. And all of all the RNA, all the translation associated RNA are giving us a bigger picture about what it is happening inside the cells and can give us insights into novel proteoform, which means also new antigen that may be detected to the translation, and novel druggable hits and biomarker development. We don't need to forget that those translation-associated RNA can also be cleaved upon a specific stress stimuli, and they can be released in the biofluids, increasing the diversity of RNA signature that can be detected. And so to capture all the dynamic of these translation-associated RNA fragments, in Immagina, we've developed our own platform that we call DORS, which is based on 3 main blocks. So we have a first block that is an omic profiling of all the RNA fragments that are present in our biological model. And then we select specific fragments based on feature selection, so such as the sequence, the structure, the presence of transcriptional modification and cleavage pattern. And this selection is done through our own artificial intelligence-driven pipeline, PROC. And after we select this feature, we can then move forward on the last step and pillar of the platform, which is the single target screening for larger validation that is based on a qPCR approach. Among all the RNA fragments that we can analyze with this platform, in the last years, we focused especially on tRNA fragments. And the reason there are mainly four reasons. The first one is for their stability, because tRNA are protected by their secondary structure and especially by the presence of several post-transcriptional modifications. They are very abundant, and they are also easier to detect than rare mutation. And they are also cancer-specific, because in different type of cancer, we can find a different cleavage pattern. And then in some of the publication, we also can retrieve that tRNA fragments may have a role in regulate gene expression and cell proliferation. So our DORS platform has been already validated in real-world patient cohort. So we had some internal proof of concept to validate the technology. The most advanced one is the proof of concept on cutaneous squamous cell carcinoma, which is a type of skin cancer, in which we already validated and selected a few tRNA fragments that are present at different stages of the disease. And now we are in the process of trying to understand if these fragments can be used to predict the response to immune therapy, in particular to anti-PD-1 inhibitors, since this anti-PD-1 inhibitor was recently approved for non-resectable CSCC patients. Besides this, in the last few months, we are also working on autoimmunity, and in particular with the inflammatory bowel disease, in collaboration with Johnson & Johnson, in which we basically established a small cohort of ulcerative colitis patients, and we will use our DORS platform to understand if we can select RNA fragments that are able to discriminate between active disease and healthy patients. And we will benchmark the data that are coming from this proof of concept with the background data set that is composed of our CSCC collected database. So, but what does it mean, all of this, for the market? So the liquid biopsies market is actually projected to grow from 7 billion in 2025 to 20 billion in the 2030. And the artificial intelligence in precision oncology is growing even faster and is projected to grow to 12 billion in only 6 years, starting from 2 billion right now. Why this matter? Because most of the liquid biopsies development at the moment are focusing only on cell-free DNA. But CF RNA is a smaller but fast-growing category that now represent only 6% of the total market, but it is projected to grow. In fact, if we look at the simple metrics, we can see that the CF DNA are actually already a crowded field in which there are more than 90 already FDA-approved tests that are based on ctDNA, while the cell-free RNA are in emerging categories, and the translational RNA fragments, where the DORS is moving, is basically an untapped space. And this is a first mover opportunity in a market that is growing at the double-digit rate. And the opportunity actually may be even bigger, because we don't need to forget that also the RNA therapeutic market is expanding very fastly, representing a $10 billion of market segment, with more than 1,000 of RNA therapies that are currently in development and 35 that are already approved. So potentially the RNA diagnostic could follow an exponential growth together with the RNA therapeutics. And so what we think is the next frontiers of RNA in liquid biopsies is actually that so far we just look at genome and then we look at the epigenome, and now we are looking at the extra layer, which is our multi-layer RNA fragmentome. So with this, I want to just summarize about what we talk about. So we begin this at the beginning of the seminar with this 90% trial failure due to lack of efficacy, and we understand that lack of efficacy sometimes means just wrong patient, and we showed how precision oncology and biomarker selection turn this around with different examples. We did some example of liquid biopsies and how those can overcome tissue limitation, with example of already FDA approved and clinically adopted test. We had an insight of a next layer of RNA biomarkers and how to detect them with our DOORS platform. But more importantly, I hope that I make it pass through that precision oncology is not just a story of biology, but is a story of biology that is enabled by technology. So at the beginning of the webinar, we say, why do cancer drugs fail? I think that now has a clear answer. They fail when we do not select the right patients. And the deeper question is if we can fix it. And the answer is yes, through patient stratification, biomarker detection, and a multi-omic profiling. And with this, I want to thank you, and I'm really happy to take questions, if any.
Maybe one question. So yeah,
oh, nice.
If it's okay. Yeah.
Yeah, of course. Go ahead.
Thank you. Yeah, I'm curious about the use of the tRNA in the liquid biopsy, because you just mentioned that you can easily find the cancer-specific cleaved pattern of the tRNA here, right? And sometimes this tRNA can also link to particular feature of the tumor, including like proliferation or something else, right? But and so can this feature can also like be linked to the particular cancer type or particular indication if they are all linked to proliferation? So can you distinguish from different kind of cancer type? So this is the first question. And the second one, because I saw your list, your table, you also target another indication, including non-oncology part. So you also mentioned about the NASH or the IBD disease. And I believe there will be different kind of pattern of the tRNA here. But in this case, what kind of biological function are linked to? So I'm sure it should not be the proliferation part, right? Because it's different feature with the non-oncology disease. Yeah, thank you.
Thank you. Thank you for the question. So regarding the tRNA fragments, basically this part, it's still in the preclinical settings, not only in Immagina, but in general, because those type of RNA fragments are emerging in the last 10 years. So there is nothing yet in clinical development. But if we base our knowledge on what is already published for cancer biology, tRNA fragments are mainly linked to cell proliferation, while for the IBD, because you were mentioning the other type of disease that we were trying to have as a proof of concept for tRNA fragments, in that case, we think that tRNA fragments are generated upon immunity cell stimulation, because in literature is reported that when the immune system is activated, it's also activated a series of nucleases that has as a target tRNA. And so there were a couple of nice work in which they introduced that upon, for instance, viral or bacterial infection, the immune system is activated, and they can see that basically there is a peak of tRNA, of specific tRNA fragments in the body fluid. So in that case, yes, it's a different type of functional role compared to cancer biology. I hope I answered your question, and I don't know if I missed something.
Oh, yeah, thank you. It's really interesting. But just one more additional question. So have you ever considered to monitor the microRNA? Because I believe it's also stable, and it can also represent the biological function or also feature in the disease microenvironment. Thank you.
Yeah, thank you. So we are not targeting the microRNA for a specific reason, because basically our platform detects specific nuclease cleavage, and this nuclease cleavage is not compatible with the microRNA biology. We think that microRNA are really interesting, and they are quietly studied in, I think, the last 30 years. But unfortunately, nothing is already yet on the market as a biomarker test. And on the other way, the tRNA fragments represent like a completely untapped area, and we are trying to move in this really highly innovative field. So
okay, thank you.
Nice, thank you. Then I think we have another question in the chat, Ale. Not sure if you can find it.
Yeah, a moment. So the question is, I'm not a biologist, but is there any famous example whether tRNA fragments detection has already proven successful application for patient selection already in clinical trials? So the quick answer is no, because it's actually an emerging field in the last 10 years. So if we think about cell-free DNA, we started to study cell-free DNA like 30 years ago and 25 years ago, and the first approved cell-free DNA diagnostic was like 12 years ago. So I think that it's too early for the tRNA fragments field to be already there. So right now there are basic research evidence that these fragments are found into the biofluid, that they can be associated with different type of tumors, but not only tumors. There are examples also, as I was mentioning before, with other type of disease like infection disease and metabolic disease. So right now there is no evidence in clinical setting, but just the preclinical one. Yeah, you're welcome.
Oh, we just said another question.
Okay, thank you for the question. So the question is the readout of tRNA fragments an absolute value, or you need a healthy control? Right now, we need an healthy control because it's a comparative analysis, but we are already working in shifting from a normal qPCR to an absolute quantification through digital droplet PCR, but we are not there yet, but we are currently working on this. Is the platform scalable in terms of number of patients? The answer is yes, because I mean, also for the omic part, we can now really lowering the input amount and using a multiplexed approach. So the platform is completely scalable for the number of patients to have a larger screening in a larger cohort. Then it depends also on if you want to, yeah, thank you.